Hey, Michelle Glass here. Welcome back to more of cardiovascular chapter. So you can see here that we've actually transitioned from our chapter 20 to our chapter 21, but I have grouped it all together on a single homework packet. Our topic for this video is capillary exchange. So remember, the whole job of the cardiovascular system is to transport either oxygen or nutrients or hormones or white blood cells or waste products like carbon dioxide gas and metabolic wastes away from tissues. So in order for there to be a purpose behind all this transport, you have to have an ability for things to enter or exit those blood vessels. And all of that entry and exit is happening at the capillary. The capillary, remember, is the vessel where exchange can occur. Now, what is actually going to regulate the exchange, or what's the mechanism of those exchanges? We have three different types of movement that's happening. We have diffusion, which we've talked about already in AMP1 to a lot of detail. We have filtration, which is kind of new, and we have reabsorption, which we'll see is actually old information, but just kind of reapplied here. Okay, so let's start with diffusion. We should already be well-versed in this. We should all know that diffusion is the movement of solute from a high solute concentration to a low solute concentration. So of course, solute, remember, is the stuff that's dissolved, and this is an automatic passive process. Now, in the case of diffusion of solute across the capillary, it is, of course, different for different types of atoms and molecules. So as we are looking at different types, if you are lipid soluble, you can go straight through the phospholipid bilayer. So if you're a lipid hormone, like we talked about in the endocrine system, if you're carbon dioxide gas or oxygen gas, these are all things that are lipid soluble. So they're able to move straight through the phospholipid bilayer. Remember, your capillary is a circle of endothelial cells. So we're talking about moving through the membrane of those endothelial cells to either go in or out of the capillary. If you're an ion and you're charged, you can't fit through the phospholipid bilayer, but you can move through a protein channel. So we will see those endothelial cell membranes will have ion channels to allow ions to move in and out of the capillary. So again, you're moving through the cell membrane of the endothelial cell to go into the cell, and then crossing another membrane to get out of the cell and out of the capillary, or into the capillary, depending on the direction of movement. If you're bigger than an ion, you may not be able to fit through the channel, or you may not have a channel to fit through. But if we are seeing these medium-sized polar molecules, such as water and glucose, it might actually be able to move through gaps in between those endothelial cells if we're in a continuous capillary. Or if it's a fenestrated capillary, you have those fenestra, those pores, that are going to be large enough for this type of molecule to move across. And then we will see if you're really big polar molecule, you can really only cross through those fenestra. So you're only able to cross through fenestrated capillaries. So we talked about continuous capillaries being more restrictive and what can cross, and that's because, again, it would have to be able to fit through those endothelial cells unless it's able to go through a channel or through the plasma membrane. When you get to the fenestrated capillary, it's a less regulated, because there, if it's able to fit through those pores of those cells, then it's able to cross automatically. The final little piece of information we need to pick up here is that those plasma proteins, namely, let's think of these as the albumin proteins, can only cross at those sinusoids. So essentially, plasma proteins and the red blood cells themselves are trapped inside of your vessels unless you're at a sinusoid. Sinusoids are, of course, located in areas where you need those proteins to enter, so that would be the liver. or you need red blood cells to enter, that would be the bone marrow, or you need red blood cells to exit when they're old and worn out, that would be the spleen. So we'll see sinusoids located in places like the liver, the spleen, the bone marrow, where you need the entry or exit of large things like those plasma proteins, namely the albumins or red blood cells themselves. Our next topic is looking at this process of filtration. Now, filtration is where you can remove solute across a porous membrane. There's not a lot of control here, although it's, based on size. So it has to be a molecule that can fit through the pore. And what we're going to see here is with filtration, there's going to be a push, shoving those molecules across the capillary. And that big push is going to be described as hydrostatic pressure. You usually just consider this to be the push from the heart pumping the blood through that arterial system. So we'll really see the hydrostatic pressure strong on the arterial end of the capillary and getting weaker and weaker and weaker as we move towards that venual end. When we talk about a filter, probably the thing we're most familiar with is a coffee filter. So, you stick your coffee grounds in and the water filters through, giving you your delicious coffee, but hopefully none of those coffee grinds or grounds are able to make their way into your coffee, right? So I have press here, coffee press. Unfortunately, it's empty, right? But we have our porous membrane, so it's a little screen there. And what you do with a press, if you've not used one before, is you add in your coffee, and then you pour in your hot water, and then you put on your press. And so the reason I'm bringing this in is because we're going to press down, forcing all of those coffee grounds in the bottom and then the coffee liquid here that we can pour out and have our delicious beverage, right? So what I want you to kind of think about is that push, that filtration. In that case, you're filtering all the coffee out of that liquid so that you're left just with the drink. That would be our hydrostatic pressure. That's our heartbeat pushing that blood through the arterial system. In the case of filtration, it's not very selective. Anything that's able to leave the capillary will leave. So if you're looking at a continuous capillary, things that can fit through those gaps between cells will be pushed out. If you're looking at a fenestrated capillary, which is more kind of what I've tried to draw here, then you're going to see stuff being pushed out through those pores. Again, the only limitation here is size. So things that are big, like those albumin proteins or red blood cells, are going to stay trapped in the blood vessel. Things that are small are going to get pushed out through those openings. So because your blood is pumping and pushing that blood through the arterial system, then stuff is going to start to leave as soon as there's openings for it to leave. But that's filtration. Again, the ability to leave is whether there's channel that's open for those ions, whether there are gaps that you can fit through between cells, or the fenestra of those fenestrated capillaries. The last thing that we need to look at here is what's described as reabsorption. So remember, reabsorption means to move back into the capillary. When we talked about diffusion and filtration, we're really talking about things exiting the capillary or waste products moving back in. In the case of reabsorption, what we're really talking about here is water and then whatever has been dissolved in with that water. So when you have that process of filtration, lots of stuff is leaving the capillary. Some of that stuff needs to be pulled back in. But remember, not everything has left. You still have albumin proteins and you still have red blood cells that are inside that capillary. So as we're moving towards that venual end, we see that the blood that is left inside is getting more and more concentrated because so much water and solute has already moved out. Reabsorption is based on principles of osmosis. So you need to be strong in that concept. You should have already mastered that concept as part of your A&P1. Osmosis remembers the movement of water from a high water concentration to a low water concentration. If you're not super confident on this topic, you do need to go back and review, and you do need to ask questions. keep in mind when we're thinking about reabsorption that not everything is leaving the capillary. Not everything can be filtered out. There is stuff that remains. We can think mostly of those albumin proteins remaining. Also, red blood cells themselves are remaining. We talked about those albumin proteins as having this role of regulating osmolarity. Here is where we see that. Those albumin proteins are basically creating an osmotic pressure that we can think of as pulling the water back into the capillary after it's been filtered out. And the significance there is water is a universal solvent. So it's not just water, right? It's whatever is dissolved in with that water is going to be able to be reabsorbed. So here we're looking at our capillary. We have our arterial end on one side, our venual end on the other. The blood pressure is highest in the arterial end because the heart, remember, is pumping the blood through those elastic arteries, and then those muscular arteries, and then those arterioles leading to the capillary. And so on the arterial end, we have a strong hydrostatic pressure forcing, filtering things out of the capillary. So again, based on its ability to leave. And then you're seeing electrolytes and water and nutrients and oxygen gas and hormones exiting the capillary here at this arterial end, going over those cells that are really close there to the capillary. Some of that fluid, notice, is actually going to be picked up by a lymphatic capillary, which we'll be talking about here in our next unit, or not unit, but our next chapter. And then as material is moving across the capillary, it's going to get more concentrated because those orange albumin proteins, they're not leaving, red blood cells, they're not leaving. So as we get closer to that venual end, we're going to have osmotic pressure pulling water in. So as we get more and more concentrated over here, we're going to see, based on the principles of osmosis, that water is going to move from high water to low water and bring with it waste material from the cell, bring with it CO2 gas from the cell, and whatever else needs to re-enter the blood here in the capillary. And the other thing that's nice here is we're creating a flow. So we're washing across the cells. It's not just like a release and then stagnant water. You have a flow of that interstitial fluid across those cells coming back into the venual end of the capillary. So we're going to call the pressure that's pulling stuff back in blood colloid osmotic pressure. The term colloid is talking about suspension of proteins. So because these proteins are too big to leave, they're concentrated, they're creating a blood colloid osmotic pressure, which is going to trigger that reabsorption of water and whatever else waste products are dissolved in that. And then, of course, that's making its way to the venule and then slowly making its way through the venous system back to the right atrium of the heart. All right, folks, that's it for now.